Assalamu alaikum, guys. Namaste. Welcome back to our lecture. Next lecture. Okay. Mhm.
Namaste,
Uh-huh.
sir.
Walaikum assalam, sir.
Okay. I think you are all well. Okay.
Yes, sir.
Uh-huh.
Yes, sir.
Okay. That's good. Okay, let's start. Our presentation, our topic today, 71 students present our lecture. Cell membrane and the transport through the cell membrane. And by the end of this section, you will be able to describe the molecular components that make up the cell membrane, explain the major features and properties of the cell membrane, differentiate between materials that can and cannot diffuse through the lipid bilayer, and compare and contrast different types of passive transport with active transport, providing examples of each. Okay? Describe differences in structure and function. All living cells in multicellular organisms have a surrounding cell membrane, as you know. As the outer layer of your skin separates your body from its environment, the cell membrane as well, also known as plasma membrane. Cell membrane separates the inner content of a cell from its exterior environment. This cell membrane provides a protective barrier around the cell and regulates which materials can pass in or out. Structure and composition of the cell membrane. The cell membrane is an extremely pliable structure composed primarily of back-to-back phospholipids, as you know, a bilayer of phospholipids. Cholesterol is also present, which contributes to the fluidity of the membrane, and there are various proteins that embedded within the membrane that have a variety of functions. A single phospholipid molecule, here you can see phospholipid bilayer, a single phospholipid molecule has a phosphate group on one end called the head, and two side-by-side chains of fatty acids that make up the lipid tails of phospholipid molecule. The phosphate group is negatively charged, making the head polar and hydrophilic or water-loving. Yeah? Have you got it? Water-loving, hydrophilic. A hydrophilic molecule or region of molecule is one that is attracted to water. The phosphate heads are thus attracted to the water molecules of both the extracellular and intracellular environment. The lipid tails, on the other hand, are uncharged. They are non-polar and are hydrophobic or water-faring molecules, water-faring part of phospholipids. A hydrophobic molecule or region of a molecule repels and repelled by water. The cell membrane consists of two adjacent layers of phospholipids. Two adjacent layers of phospholipid. You can see this slide. Phospholipid bilayer. Hydrophilic heads here, red color, and hydrophobic tails, yellow color. The lipid tails of one layer face the lipid tails of the other layer. Meeting and the interface of the two layers, the phospholipid heads face outward, one layer exposed to the interior of the cell and one layer exposed to the exterior. Because the phosphate groups are polar and hydrophobic, they are attracted to water in the intracellular fluid. Intracellular fluid abbreviation is ICF. ICF. Okay. Next, intracellular fluid. Intracellular fluid, ICF, is the fluid interior of the cell. The phosphate groups are also attracted to the extracellular fluid. Extracellular fluid abbreviation is ECF. Intracellular ICF, extracellular ECF. Extracellular fluid is the fluid environment outside the enclosure of the cell membrane. Interstitial fluid, IF, is the term given to extracellular fluid not contained within blood vessels. Not contained within blood vessels. Okay. Because the lipid tails are hydrophobic, as I said, they meet in the inner region of the membrane, excluding watery intracellular and extracellular fluid from this space. The cell membrane has many proteins as well as other lipids such as cholesterol that are associated with the phospholipid bilayer. Membrane proteins. Membrane proteins. The lipid bilayer forms the basis of the cell membrane. Its structure, okay? But it's peppered throughout with various proteins. Two different types of proteins that are commonly associated with the cell membrane are the integral proteins and peripheral proteins. As its name suggests, an integral protein is a protein that's embedded in the membrane. Embedded in the membrane. Integral proteins embedded the membrane. A channel protein is an example of an integral protein that selectively allows particular materials such as certain ions to pass into or out of the cell. This part of our topic, this part of our lecture, very significant because our next classes, practical classes, next lectures will be about transport through the cell membrane, transport different types of ions, sodium, potassium, calcium, chlorine, etc.
Yes, sir, please see. I am not somebody doing this.
Okay. So as you see, okay, turn off your microphones. Thank you. A channel protein, as I said, is very crucial part of physiology, because if the basis of physiology, transport through the cell membrane, occurring of membrane potential, action potential, and the nerve impulses, conducting nerve impulses, our next classes, it will be very significant, very useful for you. So a channel proteins is an example of an integral protein that selectively allows particular materials such as certain ions to pass into and out of the cell. Some integral membrane proteins are glycoproteins. A glycoprotein is a protein that has carbohydrate molecules attached which extend into the extracellular matrix. Peripheral proteins. Peripheral proteins are typically found on the inner or outer surface of the lipid bilayer, but can also be attached to the internal or external surface of an integral protein. These proteins typically perform a specific function for the cell. Next, membrane carbohydrates. Membrane carbohydrates, also very crucial substances of cell membrane. Carbohydrates, you see here, carbohydrates are the third major components, phospholipids, proteins, and carbohydrates. The third major component of plasma membrane. They are always found on the exterior surface of cells and are bound either to proteins or to lipids. This carbohydrate change may consist of 2 to 60 monosaccharide units. You see here, monosaccharide units, and can be either straight or branched. Okay, along with peripheral proteins, carbohydrates form specialized sites on the cell surface that allow cells to recognize each other, one of the core functional requirements noted above in cellular membranes. And cholesterol. Animals have an additional membrane constituent that assist in maintaining fluidity. Cholesterol, which lies alongside the phospholipids in the membrane, tends to dampen the effects of temperature on the membrane. Thus, this lipid functions as a fluidity buffer, preventing lower temperatures from inhibiting fluidity and preventing increased temperature from increasing fluidity too much. Okay, too much. Thus, cholesterol extends in both directions, the range of temperature in which the membrane is appropriately fluid and consequently functional. Cholesterol also serves other functions such as organizing clusters of transmembrane proteins in the lipid rafts. Okay, thank you. One of the great wonders of the cell membrane is its ability to regulate the concentration of substances inside the cell. These substances include ions such as calcium, sodium, potassium, and chlorine. Nutrients including sugars, fatty acids, amino acids, and waste products such as carbon dioxide, which must leave the cell. The membrane lipid bilayer structure provides the first level of control. The phospholipids are tightly packed together, and the membrane has a hydrophobic interior. This structure causes the membrane to be selectively permeable. A membrane that has selective permeability allows only substances meeting certain criteria to pass through in unaided. In the case of the cell membrane, only relatively small non-polar materials can move through the lipid bilayer. Remember, the lipid bilayer tails on the membrane, they are non-polar. Some examples of these are other lipids, oxygen and carbon dioxide gases, and alcohol. However, water-soluble materials like glucose, amino acids, and electrolytes need some assistance to cross the membrane because they are repelled by the hydrophobic tails of the phospholipid bilayer. All substances that move through the membrane do so by one of two general methods, one of two general ways, which are categorized based on whether or not energy is required. There are two types. First is passive transport, passive processes, or passive transport. Passive transport is the movement of substances across the membrane without the expenditure of cellular energy. Without energy is passive transport, passive processes through the cell membrane. In contrast, active transport is the movement of substances across the membrane using energy from adenosine triphosphate, ATP, using ATP, use cellular energy. Passive transport. First is passive transport. Passive transport, in order to understand how substances move passively across a cell membrane, it's necessary to understand concentration gradients and diffusion. You will study it by chemistry, these processes, and I think you studied them at school. Yes, you know. A concentration gradient is the difference in concentration of a substance across a space. Molecules or ions will spread, diffuse from where they are more concentrated to where they are less concentrated until they are equally distributed in that space. When molecules move in this way, they are said to move down their concentration gradient. So first type of passive transport is diffusion. Diffusion is the movement of particles from an area of higher concentration to an area of lower concentration. A couple of common examples will help to illustrate this concept. Imagine being inside a closed bathroom. For example, if a bottle of perfume were sprayed, the scent molecules would naturally diffuse from the spot where they left the bottle to all corners of the bathroom. And this diffusion would go on until no more concentration gradient remains. Maybe another example is a spoonful of sugar placed in a cup of tea. Eventually the sugar will diffuse throughout the tea until no concentration gradient remains. In both cases, if the room is warmer or the tea hotter, diffusion occurs even faster as the molecules are bumping into each other and spreading out faster than at cooler temperature. Having an internal body temperature around 98.6 of Fahrenheit thus also aids in diffusion of particles within the body. Next method, next type of passive transport is facilitated diffusion. Facilitated diffusion is the diffusion process used for those substances that cannot cross the lipid bilayer due to their size, charge, and or polarity. A common example of facilitated diffusion is the movement of glucose into the cell, where it's used to make ATP. Although glucose can be more concentrated outside of a cell, it cannot cross the lipid bilayer via simple diffusion because it's both large and polar. Okay, as an example, even though sodium ions are highly concentrated outside of cells, these electrolytes are charged and cannot pass through the non-polar lipid bilayer of the membrane. The diffusion facilitated by membrane proteins that form sodium channels or pores in membrane. Sodium channels or pores in membrane. So they cannot pass through the non-polar lipid bilayer of the membrane. Their diffusion is facilitated by membrane proteins that form sodium channels or pores. So that sodium ions can move down their concentration gradient from outside the cell to inside the cell. Next type is osmosis. Osmosis is the diffusion of water through a semi-permeable membrane. Osmosis occurs when there is an imbalance of solids. Don't write me, please. See, someone is kicking participants. I don't know. Don't write me, please, guys. Osmosis occurs when there is an imbalance of solids outside of a cell versus inside the cell. A solution that has a higher concentration of solids than another solution is said to be hypertonic. And water molecules tend to diffuse into a hypertonic solution. Here you can see erythrocytes and water. Water goes out from erythrocytes in hypertonic solution. Cells in a hypertonic solution will shrivel as water leaves the cell via osmosis. In contrast, a solution that has a lower concentration of solids than another solution is said to be hypotonic solution. And water molecules tend to diffuse out of a hypotonic solution, to erythrocytes. Cells in a hypotonic solution will take on too much water and swell with the risk of eventually bursting. A critical aspect of homeostasis in living things is to create an internal environment in which all of the body's cells are in an isotonic solution. Various organ systems, particularly the kidneys, work to maintain this homeostasis. Next type of transport is active transport. Active transport for all of the transport methods describes please send link of Zoom. Okay, all day I will deliver lectures for you in Cisco WebEx, in Zoom, in Jitsi Meet, then Microsoft Teams for all of you, for part of you. For Kashmiri students, they said Zoom is better. For Rajasthan students, Jitsi Meet is better. Okay, guys. Okay. During active transport, ATP is required to move a substance across a membrane, often with the help of protein carriers, and usually against its concentration gradient. One of the most common types of active transport involves proteins that serve as pumps, especially sodium-potassium exchange pump. The sodium-potassium pump, which is also called sodium-potassium ATPase, transports sodium out of a cell while moving potassium into the cell. The sodium-potassium pump is an important ion pump found in the membranes of many types of cells. Very significant part of cell membrane. Sodium-potassium exchange pump. The sodium-potassium pump is an important ion pump found in the membranes of many types of cells. These pumps are particularly abundant in nerve cells, which are constantly pumping out sodium ions and pulling in potassium ions to maintain an electrical gradient across their cell membrane. And two other types, symporters and antiporters. Symporters are secondary active transporters. Switch off microphones, please. Secondary active transporters that move two substances in the same direction. And conversely, antiporters are secondary active transport, also use energy, and the transport systems that transport substances in opposite direction. There are vesicular transport. Vesicular transport when moves larger amounts of materials into or out of cell in small membrane sacs called vesicles. There are two major categories. First category is endocytosis, and second category is exocytosis. Endocytosis by a portion of the cell is receptor mediated endocytosis is endocytosis by a portion of the cell membrane that contains many receptors that are specific for certain substances. Once the surface receptors have bound sufficient amounts of the specific substance, the receptor's ligand, for example. In contrast with endocytosis, exocytosis, taking out of the cell, is the process of a cell exporting material using vesicular transport. Many cell manufacture substances that must be secreted like a factory manufacturing a product for export. Guys, our next lecture, I will try connect with you via Zoom, because I see Zoom is better for you. I don't know. Last lecture, you asked me use Jitsi Meet, and I try today connect with Jitsi Meet, even though my teacher said me that for Kashmiri student is very bad platform, Jitsi Meet. I don't know why, because here very good extending.
Network connection, yes sir.
Good connection, no? Bad.
Because of
No, sir, this is very bad connection here.
Yes,
So actually
sir.
here we have 2G network, so that's why
2G,
it
so
does not
less
run.
than 2G actually.
Sir, 1G actually from last 2 years, sir.
1 year, yes sir.
Can you switch your cameras, please? Can you switch on your cameras? Turn on your cameras.
Yes, sir.
I want to see you. Who is speaking with me? Who is talking? Okay. Next lecture, I'll try Microsoft Teams, but I don't know if you add it to this program. We have in our university, we have Microsoft Teams, but I think you didn't add it to this program. But I don't have license to this program. I have license to Zoom program, my private license, so I can connect with 100 of you. Okay?
Oh my God.
Yes, guys.
Sir, we can't use Microsoft Teams without our student IDs either.
Microsoft Teams? Microsoft Teams?
Sir, you can make Zoom webinar.
Zoom webinar. Okay, I'll try. I will try next time. And this lecture, I send you link. Okay. And here you can change your names, yes or no? Okay guys, bye bye, see you. If you have some question, please write down to our chat, okay?